one of the benefits of publishing in JVS Vascular Science is the opportunity to be selected to present your work in webinar format. And as I'm sure all of you know, the webinar appears on our homepage, and all previous webinars are also in our online version, which is at www.jvsvs.org. So without further ado, I'll introduce our two associate editors, Drs. Gale Tang and Dr. Ulf Hedin, who are running today's webinar. And but before we turn it over to them, I'm going to ask Dr. Paul Dimuzio, our assistant editor and in charge of all of our audio visual, to give us the ground rules. Dr. Dimuzio.
Thanks, Alan. Privileged to help such a wonderful event once again for your journal. Just ask everyone if they don't mind to stay muted. And if you have any questions, please pose them to the chat, and our discussants will pick them up there and hopefully get to everybody's question. And once again, we are recording this, so I wish everybody a successful webinar. Thank you.
And thank you, Paul, and warm welcome to this here in Sweden late night webinar. We've had this a few times, and it's a real pleasure to discuss science. This time the webinar is dedicated to smooth muscle cells in vascular disease. And I have the privilege to introduce Professor Kathleen Martin, who is the Professor of Cardiovascular Medicine at Yale School of Medicine. She has a background in vascular science, doing a PhD at Case Western in '97, two postdocs at Harvard Medical School. And her focus has been for many years to investigate the regulation of smooth muscle cell function and how they contribute to normal vessel function and cardiovascular disease. So this is the theme of the webinar that Kathleen will now start to introduce to us. Thank you.
Well, I'd like to thank the organizers so much for organizing this webinar and for inviting me to present on an introduction to the other three talks based on a review article that we recently published in JVS Vascular Science called Targeting Smooth Muscle Cells in Vascular Disease. So we all know smooth muscle cells are really remarkable cells. So the smooth muscle cells that make up our arteries and veins, as you see here, even though they're differentiated, they retain a remarkable plasticity to de-differentiate, and this allows them to contribute to the growth, remodeling, and repair of the vasculature, but also contributes to cardiovascular diseases, including atherosclerosis, calcification, hypertension and pH, aneurysms, transplant vasculopathy, and restenosis. Restenosis and intimal hyperplasia is a complication of revascularization procedures, including angioplasty and stenting, where the smooth muscle cells de-differentiate and proliferate to participate in a wound healing response, but this response can go overboard and form these new lesions in the lumen. This is an example of these lesions in some rodent models of angioplasty or stenting. The smooth muscle cells normally reside in the medial layer where they're differentiated and quiescent, but in response to mechanical injury and the inflammatory cells and platelets and the cytokines and growth factors that they produce at the site of injury, smooth muscle cells will de-differentiate, lose their repertoire of smooth muscle contractile proteins, become proliferative, migrate into the lumen where they secrete excessive amounts of extracellular matrix that forms these intimal lesions, and they can migrate even through the struts of stents. So smooth muscle cells have this amazing plasticity to change their phenotype, not just a little bit, but quite dramatically, depending on the stimuli. And this is an example of some of these phenotypic switches that we've observed in smooth muscle cells. The one I'll be focusing on today is the one that's been best studied, is this myofibroblast transition that we see in intimal hyperplasia, restenosis, and fibrosis, where they become fibroblast-like, and we call this the synthetic phenotype because they synthesize so much matrix. But from lineage tracing and fate mapping studies, we know that they can also assume macrophage-like phenotype, and we now appreciate that smooth muscle cells contribute the majority of foam cells in atherosclerotic lesions. They can also become osteochondrocyte-like in calcified lesions. In severe lesions, they've even been known to modulate into adipocytes, and we also see stem cell types derived from smooth muscle cells. So a really remarkable plasticity. And understanding the molecular basis of this plasticity will hopefully provide us with new approaches for prevention and therapy, where drugs can be applied to platforms such as balloons and stents and synthetic grafts. So there are many factors that influence smooth muscle phenotype under both normal and pathologic conditions. And these include the health of the endothelium and the diffusible factors it secretes, such as NO and prostacyclin, inputs from neurotransmitters that influence vascular tone, circulating factors, including platelet-derived growth factor, which is the main one we'll hear a lot about today, that initiates lesions in the intimal hyperplasia scenario, as well as other injuries, such as from excessive lipids, toxins, reactive oxygen, and inflammatory mediators that drive atherosclerosis and other lesions. And mechanical forces and extracellular matrix contacts have very strong effects on smooth muscle phenotype, and we'll talk a little bit about this at the end. So here's another example of this traumatic phenotype from a study in our lab. This is a mouse aorta where the contractile smooth muscle cells are in green. You see this nice elongated morphology, as opposed to where this neointima is forming in response to immune injury in a transplant vasculopathy model. And in cell culture, we can see these dramatic morphologic transitions as well. This is an example of the differentiated contractile phenotype with this elongated myocyte morphology. They express a repertoire of smooth muscle specific contractile proteins, also contractile phenotype specific calcium channels and transcription factors like myocardin. Whereas in the dedifferentiated or synthetic myofibroblast phenotype, they lose this myocyte morphology and take on a rhomboid fibroblast-like morphology. They lose their contractile proteins and express extracellular matrix proteins like collagens and fibronectin, different isoforms of contractile machinery, different isoforms of channels, and transcription factors like KLF4. This is a highly simplified schematic of the transcription factors that are at the core of regulation of this phenotypic switching. So in the differentiated state, CARG regulatory elements in the promoters of contractile genes bind serum response factor and myocardin, and these complexes drive expression of these genes. Whereas with a dedifferentiating stimuli like PDGF, the transcription factor KLF4 predominates and displaces SRF and myocardin and can recruit other inhibitory factors such as histone deacetylase 2. Now, our lab has studied drug-eluting stent agent rapamycin. This mTOR inhibitor is a highly effective drug that inhibits smooth muscle proliferation, but also promotes a healthy differentiated phenotype. This is a cartoon from the first-in-man trial where this very effectively inhibited restenosis in human coronary arteries. So we're very interested in the mechanisms of rapamycin action. And these images are actually from our initial study where we showed that in the dedifferentiated state in culture, the default is for smooth muscle cells in growth factors to assume this synthetic phenotype. These are actually cells that we treated with rapamycin and could induce this beautiful differentiated morphology. And this happens at the level of contractile protein transcription. So another major drug-eluting stent agent is paclitaxel that works by inhibiting microtubules to prevent proliferation and migration. The rapamycin family of drugs inhibit the protein, the mechanistic target of rapamycin, which is a master regulator of growth in the cells. It regulates growth and biosynthesis processes, including protein synthesis. Our lab has also shown that inhibiting mTOR with rapamycin promotes an array of transcription factors that promote this contractile gene transcription, also that inhibit cell cycle machinery. More recently, we found that the mTOR pathway also regulates an epigenetic regulator, TET2, that has profound effects on smooth muscle phenotype. And for the rest of this talk, I will focus on two areas that we covered in our review, epigenetics and the hippo-YAP pathway that I find extremely exciting for future therapeutic discoveries. So epigenetic regulation is an exciting mechanism because by changing the way the DNA is packaged, the way it's wrapped around these histones in silent inactive chromatin or in open and accessible chromatin, you can dramatically alter the whole program of gene expression in cells. And so this is achieved by post-translational modifications of the histone proteins around which DNA is wrapped, or modifications of the DNA itself, methylation of cytosines. So DNA that is methylated and deacetylated is compacted in silent chromatin, whereas open chromatin is demethylated and acetylated. So DNA methyltransferases have long been known to methylate DNA and promote this compacted silenced conformation, but the TET family of enzymes were recently shown to demethylate DNA. They oxidize this methyl group into 5-HMC, and this mark is characteristic of open and accessible active chromatin. We showed that TET2 is a master upstream epigenetic regulator of smooth muscle phenotype by regulating downstream transcription factors on both the contractile and synthetic sides. So we saw that injury and PDGF through mTOR signaling repress TET2, but mTOR inhibition with rapamycin activates TET2. When TET2 is active, it promotes this 5-HMC mark at promoters, including the promoters of myocardium and SRF, the master transcription factors for smooth muscle cells, as well as at the promoters of the contractile genes themselves. Now, on the other hand, TET2 activity promotes methylation and repression of the chromatin at synthetic gene promoters, including KLF4 and KLF5, which are master transcription factors for the dedifferentiated phenotype. Now, in addition to this catalytic activity in regulating DNA methylation, we found that TET2 also influenced histone methylation marks. And we speculate that this is through interaction with other chromatin modifying enzymes. We saw that TET2 activity was consistent with other histone marks of open chromatin at contractile promoters and repressed chromatin at synthetic promoters. And this is a subject of ongoing work in our lab by Raja Chakraborty to look at how TET2 intersects with other chromatin modifying machinery to regulate DNA accessibility in smooth muscle. This is a figure from our review summarizing the major epigenetic mechanisms that regulate intimal hyperplasia. And so, as we've shown, TET2 promotes this open, active state at contractile genes. We've more recently shown that ascorbic acid, or TET2, is a co-activator for TET2. And this allows us to enhance TET2 activity and help protect from transplant vasculopathy, this other model of intimal hyperplasia. Now, other kinds of epigenetic regulators, DNA methyltransferases and histone deacetylases, have been associated with the dedifferentiated phenotype. These repressed contractile genes in smooth muscle cells, and inhibitors of DNMTs or HDACs, these have been shown to inhibit intimal hyperplasia in mouse models. Now, there's a third type of epigenetic regulator called bromodomain proteins. These are a newer class of epigenetic regulators, but these proteins are called readers because they recognize histone modifications, including acetylation, and help promote the transcriptional activation of genes. And the protein BRD4 has been shown to drive proliferation genes in smooth muscle cells. And these various inhibitors of BRD4 have been shown to inhibit intimal hyperplasia in smooth muscle. So these epigenetic mechanisms are exciting new areas that are druggable. And one of the reasons I find this mechanism so interesting is that epigenetic regulation is intimately connected to cell metabolism. In this way, you can connect the metabolic and energetic state of the cell to the DNA accessibility and gene expression program, because so many of the cofactors and substrates for these epigenetic enzymes are direct metabolites of the TCA cycle or other important metabolic processes in cells. For example, acetyl-CoA is essential for histone acetylation, and alpha-ketoglutarate produced in the TCA cycle is essential for activity of TET enzymes and Jumanji, another epigenetic regulator family, whereas the methionine cycle generates the metabolites necessary for DNA methylation. So in this way, as we know, smooth muscle cells in the synthetic phenotype have very high energy demands, and by sensing the metabolic state through these intermediates, you can connect metabolism to the cell phenotype. So now, lastly, I want to talk about the idea of cell density and cell-cell contacts and contacts with the matrix as a way to influence cell state. So the mTOR pathway regulates nutrients and energy in cell growth. It also senses growth factors, but cell density and cell contacts are another critical input for cell growth and proliferation. And these pathways, these processes together influence cell fate decisions. The mTOR pathway, which we've talked about, which is targeted by rapamycin, regulates cell growth and cell size on an individual cell level. It is activated by growth factors like PDGF and senses nutrients, including amino acids, and energy like ATP versus AMP. So injury activates this pathway and growth factors. On the other hand, organ size is regulated. The ultimate size of the organ involves the size of the cells, but primarily the number of the cells. And cell number is sensed by the Hippo pathway. It's regulated by inputs like cell adhesion and cell density. And it gets its name Hippo because it affects organ size, and Drosophila has, cell organ size phenotypes that are pretty remarkable. And this pathway, the upstream inputs to Hippo that actually sense the density are not clearly defined at the molecular level at this point, but it activates a kinase cascade that represses these transcription factors, and these transcription factors drive proliferation. So it senses density, and these YAP and TEAD transcription factors have been shown by Jiliang Zhao, who I'm excited to see is here with us on this webinar today. He's really pioneered this field, and I was pleased to write this particular editorial on his paper. He's made important contributions showing that YAP and TEAD are required for intimal hyperplasia, and that there are connections between the mTOR and Hippo pathway whereby they amplify each other's activity. Here's another schematic of the Hippo-YAP pathway from our review article. And as I mentioned, the upstream most inputs aren't directly known, but we do know that G protein coupled receptors can influence the activity of this pathway in different ways, including the prostacyclin and thromboxane receptors, which are known to regulate smooth muscle contractility and phenotype. So the prostacyclin receptor inhibits YAP signaling, where the thromboxane receptor promotes it. So agonists or antagonists of these receptors are a promising way by which we can modulate Hippo-YAP activity. And mithramycin A is an SP1 inhibitor that's been shown to influence expression of YAP and TAZ as another way to modulate this pathway and prevent intimal hyperplasia. So yet another schematic of the Hippo pathway. I wanted to include this because it shows some of these upstream inputs, such as integrins, mechanical stiffness and force, G protein coupled receptors and their ligands, and cell polarity regulators, all of which can feed into this kinase cascade that ultimately regulates whether YAP, TAZ are localized to the nucleus, where they can cooperate with TEAD to drive pro-proliferation transcription factors. So this is from the same review article, just showing the many ways, mostly indirect ways, but some direct ways by which we can manipulate the activity of this pathway, which could have important implications for modulating smooth muscle phenotype. So we'll hear more about these pathways. From Dr. Miyachi, we'll hear about Imatinib, which is an inhibitor of the PDGF receptor. From From Dr. Smith, we'll hear about a cation channel that influences contractility that reinforces a smooth muscle phenotype. And from Dr. Atkins, we'll hear about some matrix remodeling mechanisms that will influence cell-cell contacts and cell phenotype. So I'm very much looking forward to these next talks. And I just want to thank and acknowledge all of my co-authors on this JVS-VS review who are highlighted here in red. Alan, who invited us to write this article, and all of my collaborators and lab members who've made our work in this field possible over the years. So thank you for your attention.
Thank you, Kathleen. That's a very comprehensive and interesting talk, and it certainly points to the future, I think. Any questions now for Kathleen, you can post it in the chat, and I promise that Kathleen will answer you in the chat. Alan has already posted one. Okay, Gale, you can take over.
We're going to move ahead to stay on time. So I'd like to introduce Dr. Hideki Miyachi from Nippon Medical School in Tokyo, Japan, who's going to be presenting his article on Imatinib attenuates neotissue formation during vascular remodeling in an arterial bioresorbable vascular graft. Dr. Miyachi.
Thank you very much for the introduction and for reviewing and publishing our manuscript, and also allowing us the privilege to highlight our work in this webinar. Our topic is bioresorbable vascular graft. We have already succeeded the first clinical application of BVG for patients with congenital heart disease. The BVG was composed of PGA and PLCA meshed copolymer, and bone marrow-derived mononuclear cells were seated on polymer scaffold. The BVG was implanted between primary artery and inferior vena cava as fontaine operation. However, BVGs have not yet been clinically applied to the arterial circulation. According to previous studies, the mechanism of bacterial remodeling is known as follows. At first, macrophages and monocytes infiltrate into the BVG, and the macrophages release multiple cytokines and growth factors, which recruit smooth muscle cells and endothelial cells to the scaffold. Those cells are organized into a mature vessel structure. As the scaffold degrades, monocytes migrate away, leaving behind completely autologous neovessels. And platelet-derived growth factor, PDGF, also plays an important role in vascular remodeling. PDGF promotes smooth muscle cell migration and proliferation, and activates of the MMPs and fibroblasts. And Imatinib is known as a PDGF receptor inhibitor and Shikit receptor kinase inhibitor. Clinically, Imatinib is effective in treating gastrointestinal stromal tumors, cysts, and chronic myelogenous leukemia, CML. In the mouse model, Imatinib has the ability to inhibit SMC proliferation in the stenosis after repeated vascular injury. And the Shikit positive progenitor cells can differentiate into alpha-actin positive vascular smooth muscle cells. Therefore, our hypothesis is that Imatinib should attenuate nerve tissue formation by inhibiting SMC layer formation during BVG remodeling. Here is the study protocol. A total of 40 mice were administered intraperitoneally Imatinib or acetylene for 2 weeks after BVG implantation. Those mice were scheduled to be sacrificed at 3 weeks and 8 weeks in both groups. These pictures show the macroscopic findings. BVG was implanted to aura, and in a diameter of BVG is about 0.6 millimeter, and the length is about 3 meters. Results. This shows histological finding of explanted BVGs at 3 weeks. As you can see, there are almost no neotissue formation on luminal side of BVG in both groups. And behind there are some cells in the scaffold which is a macrophage as shown here. Next, at 8 weeks after BVG implantation, as shown in the left panel, control group shows a thicker neotissue formation than imatinib group as shown in the right panel. And the thickness of SMC layer and the collagen fibers in BVG were measured using image from microscopic images. The thickness of SMC layer and the collagen fiber area in the imatinib group was significantly smaller than in the control group. And the thickness of and the percent area of neotissue formation were manually measured and significantly smaller in the imatinib group than in the control group. However, the inner diameter was not different in both groups in the histology and ultrasound measurement. So as the inner diameter was similar and the thickness of neotissue formation was different. So our BVG model shows that neotissue advanced circumferentially towards the outer portion of the neotissue, neovessel. In summary, in our BVG model, the majority of cells migrated into BVG were inflammatory cells, such as M1 macrophage at 3 weeks in both groups. However, neotissue formations on the luminal side of BVG was observed in both groups at 8 weeks. SMC layers was thinner in the imatinib groups than in the control group at 8 weeks. The percentage area of collagen fiber in BVG were significantly smaller in the imatinib group than in the control group. The thickness and the percentage area of neotissue formation was significantly smaller in the imatinib group than in the control group. We propose this BVG remodeling as shown here. In our BVG model, SMC proliferates and migrates circumferentially towards the outside of BVG. But of course, the direction of neotissue formation may change depending on the materials and the manufacturing method of BVG. Our goal is to clinically apply BVG to the arterial by circulation. Thus, arterial BVG must achieve the optimal degradation optimal balance between polymer degradation and vascular remodeling within a high pressure system as arterial pressure. The result of this study can be helpful for the optimal design of arterial BVG. In conclusion, Imatinib attenuates neotissue formation by inhibiting SMC layer formation and ECM deposition into arterial BVGs. Lastly, I would like to thank Dr. Shinoka and Dr. Brewer for my support. And Angela is first in human implanted venous BVG in United States, and she's now growing up very, very well. I'm happy to take any questions. Thank you very much.
So again, if people can post their questions to the chat, I'm going to lead off with a question, Dr. Miyachi. So intimal repair and intimal hyperplasia has been shown to involve several different mitogens other than PDGF, and there's a lot of redundancy when it comes to these mitogens and smooth muscle cell proliferation. In your model, do you think that the intimal repair is really completely dependent on PDGF, or do you think that the imatinib may also affect signaling from other mitogens and receptors?
Okay, thank you very much for your questions. As you know, there are many factors involved in vascular remodeling. In this study, we focus on PDGF, but TGF-beta, BEGF, and MMP9s, and also are all well known as mitogens. In this study, imatinib attenuated neotissue formation, but neotissue formation was also observed in the imatinib group, suggesting that the other factors would have influenced neotissue formation. Imatinib also functions as a c-KIT receptor kinase inhibitor and was involved in SMC proliferation. Thus, imatinib may affect c-KIT receptor, but further research is required. Thank you very much.
So another question is that conceptually, you would assume that you really want to recruit smooth muscle cells to recreate your arterial wall within the tissue engineered graft so that it would basically replace the polymer that was originally used. So what made you decide to study imatinib in the early phase and in the center graft rather than maybe the edge of the graft where restenosis is usually more of a problem?
Okay, thank you very much for your important question. I agree the recruitment of SMC is very important and essential in early phase of vascular remodeling. But in contrast, I believe the stenosis of arterial BVG could be a problem in the future clinical applications, similar to restenosis of metal stents in coronary arteries. Thus, I agree that it's very important, very necessary, is necessary to observe it for a long time period. But our BVG was a protocol for mice, and that BVG was composed of electrospinning fibers with small porosity to withstand arterial pressure and maintain the structures. So we found that small porosity of scaffold inhibited cell infiltration and migration, and the neotissue formation was delayed. Therefore, we suggested that it's not appropriate to follow up for a long time period. But however, this study shows that Imatinib can attenuate neotissue formation, so I think it was meaningful. And the reason why we observe the central part of the graft instead of edge is because in previous study of venous graft, stenosis occurs in the central part of graft, not in the edge. The another reason is edge may cause technical problem during microsurgery, which may modify the remodeling process. Thank you very much.
Thank you, Dr. Miyachi. So we need to move on and let's introduce the next speaker of the webinar, Andrew Smith from Cleveland Clinic. His talk is on TRPC6 deficiency promotes smooth muscle cell dedifferentiation and proliferation after arterial injury. Andrew, please go ahead.
Thank you very much for the opportunity to present our manuscript on the effects of TRPC6 channel deficiency on smooth muscle cell dedifferentiation and proliferation after arterial injury. We have no financial disclosures. Calcium permeable ion channels regulate smooth muscle cell phenotype and contractile gene expression programs in a process known as excitation transcription coupling. Canonical transient receptor potential channel subtype 6, or TRPC6, is a non-voltage gated cation channel which is highly expressed in endothelial cells and smooth muscle cells, and as Dr. Martin mentioned previously, has been shown to mediate the effects of angiotensin II on smooth muscle cell contraction. We have previously demonstrated that TRPC6 mediates inhibition of endothelial cell migration by lipid oxidation products, as shown on the right. Here, wild type and TRPC6 knockout mice are fed standard chow diets or high cholesterol diets and perfusion fixed with Evans blue dye 120 hours after a carotid electrocardiography injury. Images of the explanted carotids are shown here, where the amount of blue staining reflects subendothelial collagen, thus de-endothelialized areas of the vessel. And what one can see in the wild type mice is that high cholesterol leads to an increase in blue staining, and therefore it's associated with decreased endothelial migration onto the injured segment of the vessel. And the effect appears to be blocked in TRPC6 knockout mice. The effects of TRPC6 and TRPC6 deletion on endothelial migration in terms of its downstream effects on intimal hyperplasia and remodeling after vascular intervention is not currently understood. This study was initially undertaken to determine if enhanced endothelial migration in TRPC6 knockout mice translated into decreased neointimal hyperplasia following carotid wire injury. We found several unexpected findings initially in this study that weren't consistent with our hypothesis. Here we demonstrate that there were histologic and structural differences in the common carotid arteries of wild type and TRPC6 mice at rest even prior to injury. But one can see these are 8 to 10 week old male mice, common carotid cross sections that have been H&E stained. And what one can see is that there's an increase in the luminal diameter of TRPC6 knockout common carotid arteries and a decrease in medial thickness of TRPC6 knockout common carotid arteries. It's difficult to make out on the H&E stain, but in the manuscript, we confirm this more clearly with a resource infusion stain that TRPC6 knockout media also contain a reduced number of elastin lamina compared to wild type common carotid arteries. To assess the effects of TRP6 on arterial healing after vascular intervention, we implemented the carotid wire injury of arterial injury. Here, wild type and TRP6 knockout mice were subjected to carotid wire injury and euthanized at 28 days. The injured common carotids and uninjured common carotids were then evaluated by H&E staining. What one can see histopathologically is that TRP6 knockout mice demonstrate much more severe injury phenotypes compared to wild type mice, with luminal stenosis and the accumulation of loose granular tissue or true neointimal hyperplasia. In cohorts of 9 wild type mice and 9 TRP6 knockout mice, one can see that the luminal caliber was essentially preserved after injury in wild type mice, whereas it significantly decreased in TRP6 knockout mice. We also demonstrated that in TRP6 knockout mice, there was negative remodeling of the external elastic lamina, which was not found in wild type mice. We further assessed if cell proliferation was different between TRP6 knockout, injured TRP6 knockout common carotid arteries and wild type carotid arteries. Here, H&E stained common carotid arteries of wild type and TRP6 knockout mice after injury were subjected to cell counting. We just counted the number of medial and intimal nuclei. And what we found was that there was no significant difference in the number of cells in wild type common carotid arteries, but that following injury in TRP6 knockout common carotid arteries, there was a significant increase in cell number. We further assessed if active cell proliferation was present 28 days following wire injury using immunohistochemistry against Ki67. And what we found was that only in injured TRP6 knockout mice were there Ki67 positive nuclei, suggesting that in TRP6 knockout mice, there was evidence of ongoing cell proliferation after injury. We then assessed if there was a difference in the phenotypic state of medial smooth muscle cells between wild type and TRP6 knockout mice, as our results suggested that perhaps there was an increase in phenotypic modulation and dedifferentiation of smooth muscle cells in TRP6 knockout mice compared to wild type mice. To assess this, we took common carotid arteries 28 days after injury and performed immunohistochemistry against smooth muscle actin. What one can see in these representative images is that there's essentially similar staining of smooth muscle actin in the media between uninjured and injured wild type common carotid arteries, but that after injury in TRP6 knockout common carotid arteries, there's a significant decrease in the density of staining of smooth muscle actin. And we evaluated this semi-quantitatively in cohorts of 3 wild type and 3 TRP6 knockout mice, which agreed with this observation that there was a decrease in the intensity of smooth muscle actin staining in medial smooth muscle cells following injury. These results suggested that TRP6 may have effects on arterial remodeling and the development of neointimal hyperplasia, aside from its effects on endothelial cell migration, and that TRP6 may actually have an effect on intrinsic smooth muscle cell phenotype. We therefore modified our hypothesis and posed that TRP6 is required for maintenance or re-expression of a contractile smooth muscle cell phenotype to reduce phenotypic switching and luminal restenosis following arterial intervention. That these are global and congenital knockout mice sort of impaired our ability to test this using just an in vivo model. So to determine the effects of TRP6 depletion on smooth muscle cell phenotype, we isolated, isolated primary aortic smooth muscle cells from thoracic aorta of wild type and TRP6 knockout mice. So these are just in culture, looking at these are proliferation assays of wild type and TRP6 knockout primary aortic smooth muscle cell. In panel A, cultured smooth muscle cells, these are passage 6 to 8, were plated and cultured in 10% FBS with DMEN over a 96-hour time course. And we found significant differences in the growth curve between these populations, with a significant increase in the number of cells in TRP6 knockout primary smooth muscle cells in 96 hours. B is a slightly different proliferation assay, where wild type and TRP6 knockout primary smooth muscle cells are serum starved overnight and subjected to stimulation with PDGF or 10% FBS. We found a slight, though not statistically significant, increase in the response of TRP6 knockout cells to PDGF. This is just counting cell number compared to wild type cells, and a significant increase in cell number of TRP6 knockout primary smooth muscle cells compared to wild type smooth muscle cells stimulated with 10% FBS. In addition, we assessed if migration was different between wild type smooth muscle cells and TRP6 knockout smooth muscle cells. These are razor scrape assays of wild type and TRP6 of cultured wild type and TRP6 knockout primary smooth muscle cells, and the degree percent wound healing, and thus the degree of migration, was assessed 12 hours after razor scrape assay. And what we found was that there was a significant increase in percent wound closure and cell migration in growth factor stimulated TRP6 knockout primary aortic smooth muscle cells compared to wild type smooth muscle cells. So these results in tandem suggested that proliferation and migration, or neointimal hyperplasia associated type behaviors, seem to be increased in TRP6 knockout primary aortic smooth muscle cells. We further assessed if TRP6 knockout smooth muscle cells were phenotypically different in terms of their contractile biomarkers, and if TRP6 knockout was associated with depletion of contractile biomarkers. These are Western blots of whole cell lysates of wild type and TRP6 knockout primary smooth muscle cells, passage 6 to 8. We find that there's a significant attenuation of protein expression of MYH11 and SM22 in the TRP6 knockout population compared to wild type, and we also showed this at the level of mRNA. TRP6 knockout primary aortic smooth muscle cells are associated with compensatory upregulation of closely related TRP channels, including TRPC3 and TRPC7. So thus, assessing only primary aortic smooth muscle cells, it's difficult to determine the effects of TRP6 depletion alone. The results are confounded by the potential effects of upregulation of these bystander pathways and alternative TRP channels. Thus, to determine the direct effects of TRPC6 on expression of contractile biomarkers in vascular smooth muscle cell phenotype, we performed SI-mediated knockdown of TRP6 in an immortalized murine arterial smooth muscle line, MOVAS. Here, this is 48 hours after transfection with TRP6 SIs or control SIs, and we demonstrate that in the presence of TRP6 siRNA, there was a significant decrease in protein expression of MYH11 and SM22. These results suggest that knockdown of TRP6 is sufficient to induce phenotypic modulation, and that TRP6 has a direct role in modulation of contractile phenotype and contractile biomarker expressions in arterial vascular smooth muscle cells. In summary and conclusion, we demonstrate that TRP6 knockout common carotid arteries show altered resting structure and more severe injury phenotypes in wild type common carotid arteries, and that isolated TRP6 knockout smooth muscle cells show an increase in intimal hyperplasia related behaviors, as well as a decrease in the expression of contractile biomarkers. Moreover, acute SI-mediated knockdown of TRP6 is sufficient to induce smooth muscle cell phenotypic modulation. And these results suggest that TRP6 may be an important therapeutic target for stabilization of the contractile phenotype after arterial injury to reduce neointimal hyperplasia. These are references. Thank you. I'm happy to take any questions.
Thank you, Dr. Smith. That's a very comprehensive talk. I just have one short question. Then there are more questions in the chat that I want you to answer when the next talk is on. So do you have any, very briefly then, do you have any idea of what the coupling between the channel, the calcium flux through the channel, and the effects that you see on gene expression in these cells?
Thank you. We're currently looking at the relationship between TRP6 and TRP6 depletion and the expression of master transcription factors in smooth muscle cell phenotype. We've demonstrated that TRP6 depletion is also associated with the expression of myocardin, and we're currently looking at mechanisms whereby TRP6 may modulate myocardin transcription and myocardin mRNA expression. We've shown that TRP6 regulates both the mRNA level and the protein level of myocardin. In the moment, at present, we hypothesize that TRP6 may regulate nuclear localization of the upstream transcription factor MEF2C, but obviously there are many other interesting calcium responsive candidate pathways that could contribute to these effects and could contribute to the effect of TRP6 on expression and activity of myocardin.
Thank you very much. We need to move on, but please look into the chat. There are more questions for you.
Yeah, absolutely. Thank you.
I'd like to introduce our last speaker, Robert Atkins from Nemours Alfred I. DuPont Hospital for Children in Wilmington, Delaware. He's going to be speaking on neointimal hyperplasia after carotid resection and anastomosis surgery is associated with degradation of decorin and platelet-derived growth factor signaling. Dr. Atkins.
Thank you very much, and thanks for the opportunity to present. I'm actually at the Nemours Children's Hospital in Wilmington, so pretty close to one of our moderators here. And I'm the center director where this work was done. Roshan de Cruz was the clinical fellow, surgical fellow who developed the model that I'm going to talk about, but he's back in clinical training at Vanderbilt. But this is a lot of his work. And this work exists in sort of a continuum of studies in my laboratory, trying to address interventions, surgical interventions, to improve post-surgical outcomes. We use a rabbit model for a number of reasons, largely having to do with the structure and sort of physiologic function of the adventitial tissue. And our overall goal is to be able to deploy sort of abluminally at the adventitial surface of skeletonized arteries, a synthetic pedicle. So this project is part of an ongoing set of projects involving rabbit. And what Roshan really looked at was the development of the rabbit model. And I thought we only had one slide, so I put what I could on one slide for this presentation. So the figure, the first figure is actually just showing the model that he developed. And this was a lot of work, and he developed a number of surgical models. And it's in A, it's the right, we use the left common carotid artery, and rabbits are about between 2.8 and 3.5 kilos, so relatively small vessels. And the left side, the common carotid artery is transected and then reanastomosed, and then followed with ultrasound to make sure that there's flow through the anastomotic site. 21 days after the surgical completion, the animals are sacrificed, and then we do a series of studies trying to look at the genesis of animal hyperplasia, which was not unexpected, but then linking it to pathways that we can address with some of our interventions going down the road. So what we found, what Roshan and the group found, was a distinct increase in animal hyperplasia measured at the level of the anastomotic site. Those are the H&Es in C and D that you can see here in this first part. The upper is the left common carotid, and the lower is the contralateral right common carotid from the same animal. So we used contralateral controls in this study. And then the column graph across the 5 animals that were included in this set of studies gives you an idea of what they're all significant increases in intimal hyperplasia across all 5 animals. What we're really interested in is whether or not a pathway, which is described actually by Dr. Martin and alluded to by some of the other speakers, involving Decarin and platelet-derived growth factor receptor signaling in MMP2 was associated with intimal hyperplasia in this animal model. And really what we're starting to do now is intervene in this pathway using the model. So the immunofluorescence images that I'm showing are red, green, and blue. The green is autofluorescence of elastin. The prior speaker actually talked about the elastin laminae. The blue are nuclei, and you can see just from a quick look that the total magnification on the screen is a little bit different across the 3 images. That's because of the way these images were pulled from the paper. And then the red is 3 different markers with an indication of the lumen, the intima, the media, and the adventitium by LIMA. So what we found, in addition to this increase in intimal medial thickness, was an increase in Decarin. And on Western blot analysis, the Decarin was actually fragmented in the operated side, indicating a significant change in the Decarin, not only the amount of Decarin, the location of the Decarin, but also the nature of the Decarin moiety. There was an increase in matrix metalloproteinase 2, significant increase in the intimal, in the hyperplastic zone. And then phospho PDGF receptor beta was upregulated in the intimal zone, as was phospho-MAP kinase. So the pathway that was illuminated by this links the animal hyperplasia in the surgical model with Decarin degradation and signaling through the PDGF receptor beta pathway and MAP kinases. So the progression from here, and actually a number of the things that were discussed and that we're looking at, although COVID's really slowed down some of this research, as has Dr. Cruz going back to clinical training, is trying to parse out what are the interventions that we could use and to what degree sort of the chemical and cytokine signals and the activation of the smooth muscle cells can be controlled in the animal model. So we have data, and that's going to be presented in meetings coming up, linking those pathways all together with potential interventions with our abluminal approach to providing a synthetic pedicle for skeletonized and operated blood vessels. So we think there may be therapeutic targets within that pathway, and that this model will allow us to interrogate those pathways with some ease once we can get everybody back into the veterinary ORs to do some more preparations. This work was supported by R01 to me and a number of other funding sources, including from the Nemours Foundation, where I work. Really I just want to mention his name one more time. Roshan de Cruz did the Herculean job of building the model, getting it to work and getting the surgical approach to work. These are relatively small arteries. The scale bars in those H&Es in the first and the left side is C and D. Top one's 400 microns and the lower one's 200 microns. So this was a non-trivial exercise to get this up and running. So that's the gist of the paper. I am happy to talk about any details or about how these pathways might be integrated through the systems that we're looking at, and how things are building together within laboratory using the rabbit animal model. But thank you very much.
So just a quick technical question. So when we do carotid endarterectomies, a lot of times we remove a portion of the intimal medial layer, which is supposed to also stimulate intimal hyperplasia. In your model, you mentioned that you deliberately try to touch the endothelium as little as possible. So how do you think that this model would, will relate to the actual clinical arterial intervention?
It's a great question. And the idea, which I'm happy to discuss, is that we're trying to separate what's a super complicated set of pathways, interacting pathways. So the sort of inward out, intimal to medial signaling, we understand that a little bit, I think, and from Dr. Martin's review, there's quite a bit that we might know about that. Something we don't understand as well as a community, I would say, is outside in signaling, or signals that propagate from the adventitium back through the media to the intima. So to answer your question specifically, I'm not sure how directly it reflects on common surgical procedures, but what we're trying to do is develop the model so that we can test different aspects of what's going on. So these are just transected in the reanastomose as one of multiple different surgical models that we work on in the laboratory with limited disruption, so that if there are, which we think there are outside in signaling pathways, that we have a model system that we can address those. But it's a great question. We can do that, but in my mind, I'm not smart enough to parse out all of that information that might be going on if we disrupt the endothelium as well.
So I'm just going to relay Dr. Dardik's question from the chat. So what are the advantages of this rabbit model? Should it be more widely adopted?
Those are two questions. So the advantage for us is an early interest of mine in adventitial physiology, and the vascular adventitium and the carotid artery of the rabbit is much closer to what's in humans. So we were really looking at activation of adventitial fibroblasts early on, until Roshan came in the lab with this kind of study in mind. And we can't do that in smaller animals. So the big idea was that the rabbit is small enough, not super expensive, not all the way up to porcine or canine models, so that we could crank out a number of surgeries. So in that sense, I think it is a very good model. It is a tricky model. The vessels are still small, even the carotid's not for the faint of heart, getting in there surgically. And some of the genetics and some of the probes that other labs are able to deploy fairly readily are difficult in rabbit. So I would say it is well worth using, but to really understand what the model's strengths are. And Dr. Tang asked the question about, well, we don't disrupt the intimal layer, because we're really trying to understand that adventitial influences on animal hyperplasia and on the pathophysiology in the vessel wall. So yeah, if anyone's interested in using rabbit in the lab, I'd be happy to talk to them about our learning curve over the few years that we've been working with them.
So Dr. Hedin had a question about whether you see the same effects on Decorin in a regular balloon injury model as opposed to transection and reanastomosis.
Yeah, I think in other studies, we haven't looked in our lab, but in other studies, the expectation is you see similar pathways in a balloon sort of damage based on extension of the blood vessel from a balloon. So we weren't surprised that this pathway was activated, but we needed to establish that it was, and at least look in our simplified model that the Decorin pathway, and especially the phospho-PDGF are through MAP kinase, that's the pathway that we should be targeting, or that we could be targeting going forward.
Thank you very much. I would really like to thank all of the presenters for the wonderful research that you've presented. A big thank you to Dr. Martin, who's our featured speaker. Thank you so much for joining us. And this was really a fantastic, successful webinar. Please visit www.jvsvs.org, our journal's homepage, and look forward to seeing you at our next webinar. Thanks for coming.